Hello and welcome again. So now today we will be moving forward to our next chapter where we have discussed about the innate immune response, their components. Now we are going to meet the adaptive immune response. So how the innate immune response has taken the handle the situation of the infection that has breached your anatomical and physiological barrier, which we have discussed previously. Now, if the innate immune system is not able to control the infection by itself, then the cells, the part of the innate immune system, or say the cells of the immune system, that is the antigen presenting cell like macrophage, dendritic cell, they will take that message to the adaptive immune system and deliver the message that now the innate immune system is not able to handle the situation, not able to control the infection, you need to take over. And so the first thing is that the antigen presenting cell will first try to clear the infection, they will capture the infection, but if they are not able to do, then they will obviously then switch into the presentation and then goes to the localized lymph node, and then they activate the respective T cell, and then the T cell will activate the B cell and antibody will form, and again they will come back to the infectious site and they'll try to clear the infection by the antibody, that is activation of the complement, opsonization, clearing of the immune complex, and by other means the whole power of immune system is thrown in the site of the infection and try to clear the infection. Okay. So we are talking about the secondary lymphoid tissue, that is the innate immune response meets the adaptive immune response. So in this chapter we'll only talk about how the innate immune response will meet the adaptive response. In the next chapter, the chapter 6, we will discuss the activation of T cell and B cell. But here we will only discuss about the understanding how these cells go and present to the adaptive immune system, that is T cell and B cell. Okay. So let's move on. So we will be learning objective will be demonstrate the understanding of inflammatory response and solve the problem concerning structure and migration of to secondary lymphoid tissue. So the migration to the secondary lymphoid tissue, if we are talking about within a few hours of initiation of acute inflammatory response. So within a few hours of this initiation of acute inflammatory response, what will happen? The professional APC, that is antigen presenting cell, that have phagocytosed and processed the invading antigen begin to leave the lymphatic area via the lymphatic vessel. So what is happening? The inflammation has begun. There is a breach, there is an infection in your skin or any part of your body. The inflammation has begun, and the antigen presenting cell, professional antigen presenting cell, that is the dendritic cell and macrophages, they phagocyte and process the invading antigen and begin to leave the area and goes to the via the lymphatic vessel. The dendritic cells are probably the most efficient. They are the most efficient cell, these cells, and retract their membranous process to round up, and they begin the journey to the closest lymph node. So they will go to this closest lymph node. What is happening? The inflammation has begun, they are phagocyting, they are trying to kill the infection, clear the infection. If they are not able to do, then this dendritic cell, the antigen presenting cell, they have phagocytosed, and then they are taking that phagocytosed this material to the closest. They are leaving the infection area and going to the closest lymph node. Okay. As discussed earlier, dendritic cell and other phagocytic cells such as macrophage bind to the antigen via this pathogen recognition receptor and with a limited diversity such as this toll-like receptor. So we know this dendritic cell and macrophage bind with the antigen by this toll-like receptor. The activation of this toll-like receptor induces an acute inflammatory response in this tissue, leading to the production of pro-inflammatory cytokine. This cytokine causes a change in the phenotype of the phagocyte, which eventually alter the migration pattern and enhance their function. So this we have discussed in the acute inflammation, where we have known that when the cell goes for the phagocytosis, they release the cytokines. The cytokines comes to the back to the blood vessel, where they change this pro-inflammatory cytokines cause the change in the phenotype of the phagocytic cell. So the phagocytic cells goes under the modification. In this endothelium, like you can say mucin-like camp or integrin comes phenotypically expressed on their cell, and they bind with the integrins on the endothelium and transmigrate and then extravasated from the blood vessel and goes to the infectious site, which we have discussed. This activated dendrite cell will begin to express a chemokine receptor called CCR7, this CCR7 is activated by the chemokines that are produced by the endothelium. So this actually dendritic cell will express a chemokine receptor called the CCR7. Okay, and this CCR7 is activated by the chemokines that are produced by the endothelium. So this is activated by the chemokine produced by the endothelium. The chemokines bind to the CCRC and the dendritic cell allow them to exit to the tissue. So they will allow this dendritic cell to exit the tissue. Upon activation, dendritic cell will focus from antigen capture to the antigen presentation. Initially, they were actually thinking of capturing the foreign invader, the bacteria, the antigen only. But once they are activated, this dendritic cell, they will now convert from capturing to the presentation and then goes to the local lymph node. So upon activation, this dendritic cell, upon activation, what is happening? This dendritic cell will switch the focus from antigen capture to antigen presentation. Activated dendritic cell concentration in the draining lymph node. So the activated concentration in the draining lymph nodes and become trapped in the paracortex. So what is happening? You have known there is the infection at the site, where they from the site the dendritic cell has captured that bacteria. Now initially they captured, now they are presenting, they are leaving that infection area behind the lymphatic vessel through the receptor that is CCR7. They are leaving that area, enter into the lymphatic vessels and going to the where? Going to the local lymph node, where in the local lymph node this antigen capturing has switched to the antigen presentation, and their concentration becomes high in the draining lymph node. Mainly they are trapped in the paracortex. So the paracortex is area where T cell is present in the lymph node, and now this your antigen presenting cell has reached to your lymph node. The T cell has reached to the T cell in the lymph node, and now they will show what they have captured and then present to the T cell, and the T cell will be recognized that cell, and then T cell will be selected and they will be activated, and they will release this chemokines and then will activate the B cell, and the B cell will convert into plasma cell, and then antibody will form. So this all are the process. So this is the details of how your dendritic cell, your antigen presenting cell, leaving the site of infection towards through the lymphatic vessels and reach to the lymph node into the paracortex region where the T cell is present. The naive T cell expressing CCR7 bind to the chemokines on the high endothelial venules and then migrate to the paracortex. So there is the naive T cell as well as dendritic cell with the antigen presentation. So they will both now interact each other. One thing is important, that considering the vast number of the pathogen that enter the body, it would be nearly impossible task for the lymphoid cell to travel all the body side to protect the host. So thinking that this is a very true thing, that the lymphoid cell like T cell and B cell can't move all around the body to see where is the infection. You may have the 2 or 3 infection at a time. You may have a different infection and different site of body. So and they cannot travel each and every place in your body. So it's the role of the antigen presenting cell to capture the infection from the different parts or different site of the body and then bring to the local regional lymph node, and that is feasible. So thus the antigen are taken to the secondary lymphoid tissue where the lymphocyte constantly recirculate in order to come in contact with their specific antigen. So it's the role of the antigen presenting cell to take to the secondary lymphoid tissue, and which is feasible thing. Okay, so we have known that obviously the antigen presenting cell, the T cell will not come to the periphery, it will remain in your lymph node, and it's the responsibility of your antigen presenting cell that's to take the infection information towards the regional lymph node. If the initial tissue damage is sufficient, these cells can also flush into the blood vessel, ultimately becoming trapped in the vascular sinusoid of the spleen. So one thing is important to understand, that there are the 2 places where this T cell can be found. One in the lymph node, and another is if it is in the tissue. And if you are talking about the blood, if the infection is in your blood, then they will be carried to the spleen. So in the spleen there is the T cell, where you can think like for tissue the lymph node is blood vessel, for the tissue the lymph node, the T cell is present in the lymph node. For blood vessel, for blood the T cell is present in the spleen. So in any infection, any antigen present in the blood will be carried to the spleen where the T cell is present. Okay, and in case of tissue, we know through the lymphatics it will be carried to the lymph node in the paracortex region. So if the initial damaged tissue is sufficient, this can be flushed into the blood vessel, ultimately becoming trapped in the vascular sinusoid of the spleen. Regardless, the secondary lymphoid organ, that is lymph node and spleen, are the site where the naive mature lymphocyte will be first exposed to the specific antigen. So where is the site where the first antigen is exposed to the adaptive immune system, and that is the lymph node and spleen are the site where the naive mature lymphocyte will first expose to the specific antigen. So adaptive immune system will first encounter the antigen in the lymph node or in the spleen. Okay, so this is the picture where you can see from the site of infection, if it is a dendritic cell associated antigen, if it is the tissue area, then that will be through the lymphatics, will carry to the lymph node, the regional lymph node, through the afferent, and exits to the efferent lymph node. This lymph node is lymph is actually carried by the thoracic duct to the inferior vena cava, to the vena cava, and finally to the heart, into the blood. And if the pathogen is in the blood vessel, they will be carried, the antigen transferred by the lymphatics into regional lymph node or via the blood to the spleen. So if it is in the blood, they will carry it to the spleen. So that you have to understand. If it is in tissue, that will goes to the lymph node. If it is into the blood, they will carry it to the spleen. Now let's discuss some of the internal structure of the spleen as well as the lymph node where the T cell is actually present. So this is important because when we have an infection, what happens? This T cell area become active and they become hypertrophic because their activity is increased. Why? Because obviously the antigen has been captured and presented in the T cell area. So T cell area become active, and that you have to understand. So where the T cell area is located, which is the B cell area, is important to understand the concept of hyperplasia occurring or activity occurring inside the lymph node, or say in case of the spleen. So structure of the secondary lymphoid tissue, lymph nodes are the small nodular aggregate of the secondary lymphoid tissue found along the lymphatic channel of the body. They are designed to initiate the immune response to the tissue-borne antigen. So we know for the tissue-borne antigen, the lymph node are the place where this reaction takes place. Each lymph node is surrounded by the fibrous capsule that is punctured by the afferent lymphatics. The entry point is afferent, which brings out the lymph to the subcapsular sinus. The fluid percolates through the outer cortex area that contains aggregate of these cells called follicle, and the lymph then pass into the inner medulla and then the medullary sinus before leaving the node through the hilum in the efferent and lymphatic vessel. So afferent is enter, efferent is exit. Ultimately, the lymph node through the body is collected into the thoracic duct, which empties into the vena cava, returns to the blood. So this is the one of the way of the structure of your lymph node. Let me show over here. This is your afferent lymph nodes. Here is the capsule. Then there is the this cortex. Then this is will be the paracortex region, and there is the medulla. In the cortex region, there is the, you can see this is the primary follicle, that is B cell rich. Cortex region is over here, okay, and there is the paracortex region as well, which is the T cell rich region area. So we have to remember this thing. Only to this is very important to understand, to remember where the T cell is located in the paracortex of the lymph node. Where B cell is located, they are located into the primary follicle, that is the cortex region of the, that is the B cell rich. That is, you have to understand. So there is the cortex, then paracortex and the medulla. From the fluid enter from here, goes into the paracortex region where the T cell get encounter, and they will process and present and then further move the reaction of antibody production. This is the exit site is efferent lymphatics. Talking about this is the place, you can see the dendritic cell, the T cell area inside the lymph node. This is the macrophages, and the same thing is, so bacteria is engulfed by the macrophages, which has been shown over here. Now we will talk about the spleen. So talking about the spleen is also a secondary lymphoid organ that initiates the immune response to the blood-borne antigen. Tissue-borne, lymph node; blood-borne, obviously spleen. So if we have a splenectomy patient, then any infection that is present in the blood becomes a recurrent infection, and we need to vaccinate that patient because there is no immune protection inside. If any antigen, if any bacteria, any viruses enter inside your blood. So in that situation, a splenectomy patient is susceptible to many infections because the blood-borne pathogens are actually dealt by the spleen. So spleen is a secondary lymphoid organ that initiates the immune response to the blood-borne antigen. A single splenic artery enters the capsule to the hilum and then branches. They are divided into the arterioles, which become surrounded by the cuffs of lymphocyte known as this periarterial lymphoid sheath. So this you have to remember. This is simple. In a spleen, one artery get enter splenic artery that break down in branches into the arterial, which becomes surrounded by the cuffs of the lymphocyte known as the periarterial around the arterioles, that is lymphoid sheaths, and that is called the PALS. So this is simple. Around the arterioles there is a lymphoid, the T and B lymphocyte is there, mainly PALS, where is the T lymphocyte is there. So PAL is mostly T lymphocyte, that is the periarterial lymphoid sheath, where the T cell is present and B cell is present inside the corona. So this is the transverse section, and this is the longitudinal section where we can find the place for the T cell and B cell. This is B cell area, this is B cell area, this is T cell area. Okay. So lymphoid follicles surrounding by a rim of lymphocyte and macrophage are attached nearby. This constitutes the white pulp. The arterioles ultimately in the vascular synods, which make up the red pulp. So there are the white pulp and red pulp. Red pulp area is the arterioles area, and the lymphoid follicles surrounded by the rim of lymphocyte and macrophage are attached nearby. This constitutes the white pulp. So white pulp area is the actually lymphocyte and macrophage containing area. So white pulp, there is a in the white pulp, lymphoid follicles surrounded by the rim of lymphocyte and macrophage are attached nearby. This constitutes the white pulp. So white pulp is composed of the lymphocyte and macrophage, whereas the red pulp is the arterioles ultimately in the vascular sinusoid, which make up the red pulp. So from here, venules collect the blood into the splenic vein, which empties into the portal circulation, and from the portal circulation goes to the vena cava, inferior vena cava. So this is the transverse section, and this is the longitudinal section of your area, where you can see this is the marginal zone, this B cell corona, germinal center, PALS, central arterial, marginal sinus, and red pulp. So there is a white pulp and red pulp. Red pulp area is for the arterioles, white pulp area is for this lymphocyte and macrophages. So in the white pulp area, you can see there is this area where there is a B cell corona, and this is the PALS, where is the T cell is present. In the longitudinal section, you can see this is your PALS, this periarterial lymphoid sheath. This is the arterial, and where is the periarterial lymphoid sheath contain T cell, and this is the actually B cell area. This is the B cell area. Okay, the B cell corona. So we have understood where this B cell and T cell is present inside the lymph node and the spleen, which is responsible, which is the first place to encounter this antigen by the adaptive immune system and then process further. We'll discuss about how they are processed in exogenous material and endogenous material in the next consecutive lecture. Thank you.